Thank you for your interest in Cytophage Technologies poster titled Phage Training an Enterotoxigenic Escherichia coli bacteriophage presented at the Porc Show 2021. Enterotoxigenic Escherichia coli, also referred to as ETEC, is one of the leading causes of bacterial disease, resulting in significant economic losses in the swine industry. We are all very aware and concerned about antibiotic resistance in the swine industry in Canada. Currently, swine producers have limited alternatives to control bacterial disease, which is where bacteriophage or phage therapy can help. Phages are highly specific viruses that infect and destroy bacterial cells. The specificity of phages is important to help prevent the disruption of the existing gut microbiota. Cytophage Technologies Incorporated is using trained phages to target bacterial infections of concern for local farmers. Today I will take you through the work Cytophage Technologies is doing to train a phage to target a disease-causing strain of ETEC known as CIB54 that was isolated from a Manitoban farm. At Cytophage, we have isolated a library of phages and tested their natural host ranges so that we know what bacterial strains they can target. No phages were found that targeted this strain. Therefore, we used phage training to improve the ability of a phage to target this strain. First, a natural Escherichia phage was passaged repeatedly on the ETEC strain of interest, and every third passage was tested for the ability to form plaques, which are areas where the bacteria have been destroyed. After 3 passages, plaques were formed at high concentrations, approximately 10 to the 9 plaque-forming units per milliliter. And by 12 rounds of passaging, the plaques were much clearer visually, indicating strong lysis of the bacteria. As you can see here, the untrained phage was unable to form visible plaques, but the trained phage was able to form isolated plaques. This ability to form plaques is extremely important for the isolation, purification, and quantification of phages, which is necessary for therapeutic dosing. The 12th passage then underwent plaque purification to ensure a homogeneous population of phages. Using electron microscopy, the newly isolated phage was characterized and classified as VB EcoM CIP75, or CIP75 for short. Each round of passaging and the final trained phage were sent for sequencing to identify any potential mutations that could be responsible for the ability to target CIB54. Sequencing revealed no genetic mutations in the 6th passage, indicating that genetic mutations were not required to target CIB54. Single nucleotide polymorphisms, or SNPs, are single base pair changes in the genome. SNPs were identified in passage 12 and the purified CIP75 phage, but only one, shown here in red, was the same between the 2 phages, suggesting that the other mutations are likely side effects of the prolonged passaging, but that the mutation that is present in both may be involved in the improved plaque formation that was observed. Future work will be needed to determine this. Next, we are evaluating the efficacy of the trained phage using the waxworm model called Galleria melanella. We will have 3 groups of waxworms. The first group, the control group, are injected with the CIP75 phage alone and then observed for 24 to 72 hours to ensure that the phage are not causing detrimental effects to the larvae. The next group is infected with the ETEC strain CIB54, followed by treatment with saline solution. And as you can see here, after 24 to 72 hours post-infection, the larvae would succumb to the bacteria. In the final group, they are infected with the target bacteria and then followed with the phage treatment. By 24 to 72 hours, if the phage is effective, we will have the majority of the larvae healthy and alive. This waxworm model is a proof of concept for safety and efficacy of the phage treatment prior to the large animal trials we will perform in piglets. In conclusion, the training of natural phages allows for the improved targeting of a disease-causing ETEC strain in vitro. In vivo assays in the waxworm model will guide the design of our swine trial, where the efficacy of this trained phage will be tested on piglets. Cytophage Technologies wants you to consider that trained phages offer a safe alternative to antibiotics to treat and prevent bacterial infections, keeping your livestock safe and profitable. Thank you for your time.